Hi everyone. It's a pleasure to be able to join you today to talk about morphological phylogenetics. This talk is covering a variety of different topics, but mainly trying to focus upon some of the current controversies and perspectives with morphological phylogenetics on a general level, rather than a particular sorting out relationships with a particular clade, and some of the approaches that might be taken to try and investigate the nature of morphological phylogenetics and morphological data, and what we can do moving forward. So this is going to involve looking at simulations and empirical data being used to evaluate morphological phylogenies. So I guess we could be described, although this is perhaps controversial for this audience, as living in an age of molecular phylogenetic supremacy. Genome level scale phylogenetics has never been more practicable. They are now almost the standard for lots of morphological phylogenetics. And at the same time, there's a long existing battery of sophisticated models that we have. They're ever improving and they're ever evolving in themselves. These evolutionary models are evolving in the sense that we understand the theoretical and practical way that molecular data evolve. So whilst that's been going on, morphological phylogenetics could be described as maybe not undergoing that same level of revolution. And the proportion of morphological data that are used in the phylogenetic studies is going down, as indicated by this level of Lee and Palchi as proportion. And that's mainly as a result of the increasing vast abundance of molecular sequence data going on. However, as was demonstrated just by the previous talk by Jorge, morphology remains essential. And I don't think I need to convince anyone in this audience that the morphology remains essential. Not only is morphology an important and valuable source of information to help us reconstruct evolutionary history, but it's also essential for allowing us to integrate fossil taxa to unlock the rates and sequences of phylogenetic evolution, because morphology is the only source of data available for fossils in the vast majority of cases, as was as demonstrated quite deftly by Jorge in the preceding talk. This might lead us to a fairly big question of what is the most accurate method for morphological phylogenetics? How should we be using and applying morphological phylogenetics? Because whilst we've had this molecular revolution in terms of how we might deal with the scale and sophistication of data, there are still some very basic questions about morphology that haven't been answered with the same level of confidence. So, for example, we might consider parsimony as the traditional way that we've analyzed morphology throughout time, where we might try and reconstruct or prioritize the simplest solution, the solution that makes the requires the fewest number of character changes. Alternatively, we begin to see an increasing use of Bayesian techniques, where we might use some evolutionary models to try and consider the probability of our trees in relation to priors and models. And these two different approaches have been used to varying levels of success with morphological phylogenies. Part of this ambiguity over which might be better comes in part from simulations, where we've started with a situation where we try to evaluate morphological phylogenetic techniques. The same principle applies to molecular phylogenies. We might start with a known tree, some sort of topology of a tree. We simulate characters onto that tree using some evolutionary model or non-model or randomness, and depending on the perspectives taken, and then infer topologies from those characters using different methods, and then evaluate the performance of those methods in terms of the tree that results from those characters back against the known starting tree. Just as an example in this, Wright and Hillis was an important contribution in this. April Wright published a paper where they simulated some characters based on known tree topologies using the Jukes-Cantor model, and came to the conclusion that Bayesian techniques were outperforming parsimony. This led to quite a lot of different discourse, shall we say. Part of the problem might have been highlighted there was that the models that they were using to generate the data were very similar to the models that were being used in the Bayesian techniques to evaluate that data. So certain attempts were made to escape that possibility by looking at some teams from Bristol who have considered different ranges of models under HKY scenarios and different ways of generating that data. Alternatively, Golubov and others have considered relatively model-free methods, where they've taken an attempt to generate data based on distributions of empirically observed homoplazy, and came to the conclusion instead that parsimony was creating these more accurate results. Now, this isn't an exhaustive list, but this gives you a sort of current state of play of the situation, where there's been a variety of different ways that methods have been, simulations have been used to generate phylogenetic data, to evaluate different techniques, and have reached various conclusions about what might be the best way to do that, and various different metrics that might use to evaluate the phylogenetic performance of different evolutionary methods. So in this talk, I wanted to ask the question, what is the best way to build accurate phylogenies from morphological data? So spoiler alert, we may not land on one specific answer there, but also where we ask the question about how we might approach morphological data moving forward in the future. So in the first part, we're going to have a look at some evolutionary informed morphological simulations. We're going to consider using molecular benchmarks to try and evaluate morphological data, look at the circumstances and the relationship between morphology and molecules, some unpublished research, and then looking at some stratigraphic age range data as another benchmark to try and evaluate empirical real-world data. So in the first instance, I mentioned some of those examples where data have been generated to evaluate the performance of phylogenetic methods in simulations. Most of those were based on molecular-like substitution models on fixed trees, as was Wright and Hillis and O'Reilly et al. And Patek et al. But a variety of different ways. Another one is to assign characters to nodes in a fixed tree, which was by Goluboff et al. And Patek et al. And a variety of others, where the tree was used to generate those data using different kinds of approaches. We took a different approach in our paper, where we were just trying to consider evolutionarily informed models where we simultaneously derive the tree and the characters in evolutionary simulations. So under these circumstances, the tree topology is intrinsic property of the simulation, and the trees and the characters evolve and are derived simultaneously. So rather than starting out with a tree and evaluating characters onto that tree, we start with a system where we generate that tree and character together. So this was a project led by Joe Keating, a PDRA, working in my lab with some other collaborators in Manchester and in London, where we tried to generate some phylogenetic data in a morphological style from two different types of models. We used a species birth-death model, where we have lineages evolving randomly through time, which we might describe as a drift-like model. We've also got this new program that Russell and Joe together we wrote, was TrivoSim, where is an individual-based model, where we have a series of digital organisms that compete with each other in a digital environment, and they are given a certain probability of breeding and succeeding over many generations, and then they have a certain probability of speciating as they accumulate certain numbers of character differences. So the important thing about this one is that it's individuals in this digital simulation, and those individuals undergo a process of natural selection. I don't think I need to go into the details necessarily of how that model works, but it's there if you wanted to quickly consider that, where we consider basically how these digital organisms are competing and in terms of their fitness to an environment which can change and drift through time. So the data generated are quite different from each of these models. We tried to assess them in a wide variety of different metrics, based on not just on homoplasy, but excess steps, dispersion means, and morphological coherence, which was based on how well the data reflect the tree topology, the relationship between branch lengths and character differences, and also tree shape. So we see there's quite a range of data presented by these two evolutionary models. So we subjected those data to parsimony and Bayesian investigations and tried to ask the question of which of those were producing trees that were more similar to the original true tree. So here we've evaluated the standard consensus methods for those trees. So here in purple is the 50% majority rule consensus tree, the standard method of consensus for Bayesian trees. And here are the strict consensus of the most parsimonious trees for equal weighted parsimony and implied weighted parsimony. And generally speaking, those Bayesian posterior distribution and majority rule consensus trees are more accurate, but they're also less precise. So in some senses, we've got to remember that the consensus trees under these circumstances are doing different things. Also, I should mention this is true of the variety of different metrics we've used to evaluate this: SBR, TBR, quartets, and Robinson-Fultz distances. So we need to consider the fact that the consensus trees for a Bayesian search and the consensus trees for a parsimony search are attempting to do different things, really. the 50% majority rule consensus tree is relatively conservative in comparison to some of the implied weighting parsimony tree, which is generally quite precise. So we've got to weigh up this precision against accuracy situation. When instead we try to compare more strictly like for like trees, where we consider fully resolved trees, under this circumstance, the fully resolved Bayesian posterior distribution trees, fully resolved parsimonious trees, or fully resolved Bayesian maximum clade credibility trees, we find the results are much more similar to each other, apart from in the drift-based model. So in this situation, we might want to therefore move beyond the idea of looking at the consensus trees. And we need to remember that we need to compare like for like. For example, Martin Smith was expanding the idea that maybe some bootstraps with implied weighting or equal weighting much more comparable to bootstraps with the Bayesian phylogenetic derived tree. We have to make sure that we're comparing the same thing. The other important finding from this was that we have a new model that explicitly incorporates selection, natural selection. And this is some unpublished work where we try to consider the range of values in terms of the homoplasy and tree shape that are generated from these different kinds of evolutionary simulations that include selection or no selection. And we see that they occupy a wide range of different sort of homoplasy and tree space, which is sort of theoretical PCA construct in this idea, with empirical data empirical real-world morphological data existing over a range of things that are more consistent with molecular substitution models or selection-based models or non-selection-based models. But I'm not going to go into much more detail about that, because in some senses, we need to move on to this idea that we're thinking about simulations is very useful, but it's also constrained by some very real-world problems, in the sense that simulations are an oversimplification. They're an idealization of how we think evolutionary processes should work. But the real world data is actually comprised of much more tricky and unaccounted for aspects in it that we need to think about when we're evaluating morphology and morphological techniques. I've given this slide the title, "The Real World is Ugly," which I think is a bit harsh, a bit nihilistic, but this is just an example of some of the real world complexities that arise from real morphological data. This is an interesting example where we might come up with this hypothesis that morphology is oversaturated from Davalos et al. This idea that we've over-collected morphological characters and we're duplicating things, which relates to the idea that we might have character non-independence. Maybe we're contravening one of these core assumptions when we consider this example from mammal teeth, whereby small genetic changes can lead to massive knock-on effects of interrelated morphological characters. Or another example from mammal teeth, we've got ecological linkage. Maybe the morphology that we're observing isn't actually detecting evolutionary history, but some sort of ecological adaptation, like diet. Or in some of the earlier work that I might mention very briefly later on, there are subdivisions of morphology that show some different patterns depending upon the kinds of things that they're related to. So under these circumstances, given the real world complexity of data, we might want to try and analyze what's happening in the real world. The difficulty arises that we might not have the same level of benchmarks. In those simulations, we have those known true trees, but we don't have that for real world data. So we took an approach to try and evaluate morphological data, real world empirical morphological data against some benchmarks, and we tried to compare the real world morphological data sets inferred under both Bayesian and parsimony circumstances and compare that against a molecular topology. Now, this allows us to do two things: not only consider the properties of morphological data and morphological techniques, but also move beyond that and start thinking about the comparisons between morphology and molecules more generally. So we took an approach of compiling combined morphological molecular data sets from the literature, inferring parsimony and Bayesian trees, and then measuring the differences between those in combined and individual data sets. This is a study, again, from the same project with Joe, the PDRA working in my lab with Russell Garwood, which is currently in review. So let's have a look at some of these results, shall we? So we have a meta-analysis of 32 empirical, molecular, and morphological data sets. I don't know if there's anyone in the audience that's related to some of those. I think there might be at least one or two. Of a combined total of over 1,000 taxa and 8,000 plus morphological characters. So from those, we derived Bayesian and parsimony trees of the morphology and molecular trees as well from molecular data, and combined trees using both the morphology and the molecules. So bearing in mind that caveat that we need to move away from and only looking at like for like, when we consider fully resolved trees of the same number from all the 32 data sets, so this is where we've selected the most probable, the trees from the Bayesian posterior distribution that have the highest probability to the same number as the equal weighted parsimony. We see that they occupy almost exactly the same distribution with respect to their congruence with the morphology and molecules. So there's very little differences there when we're comparing like for the parsimony and Bayesian trees with respect to their morphological and molecular congruence. There are differences when we look at those consensus trees, in the sense that the consensus trees of the mature Bayesian inferences are more conservative and therefore have fewer inaccuracies. But again, we're not comparing like for like. When we compare like for like, there's very few differences. However, this allows us to do, which maybe should move away from these raw metrics of accuracy, in this case quartets or Robinson folds, and instead start thinking about the visualization of how these results differ in tree space. These results might be a bit abstract, so let's focus in on one example where we can find, this is an MDS visualization of the tree space occupied by the results from one data set under different kinds of inference. So over here in the sort of pinky crosses is the Bayesian the Bayesian morphological results over here. So they're occupying quite a diffuse range, as we would expect, because this is the posterior distribution. And unlike the parsimony results in both this triangle and this circle, which are much more constrained in the here we go, that might work. The parsimony results here, which are much more constrained. We've got this much more precise result where there's fewer trees as opposed to the posterior distribution. Over here in blue is the molecular results, and over here is the combined results. So what's cool about this is that we are using the visualization to see how this works on all 32 data sets. But one of the pictures that comes up is obviously this expectation of precision and accuracy being different depending upon the type of inference method, but also that the combined analysis is occupying a unique part of the tree space in almost all these examples. The combined morphology and molecules is picking up something that's different from either the individual morphology or the individual molecules. So for this first part then, this study is, some minor conclusions for this part is that the combined analysis is sampling unique tree space. Interestingly, when we found using Bayes factor analysis that the molecular and morphological partitions are not combinable and are unlinked. So there are significant differences in the types of evolutionary signal being picked up by the both of them. This isn't just about partition size. So it doesn't seem to be the case that you can swamp out the morphological signal by adding more and more molecular characters. There doesn't seem to be that relationship. The molecular data and the morphological data have a combined input, no matter how much or how big those partitions can get. Although obviously there must be a limit there. So these observations together underscore the importance of morphology and say that we shouldn't be neglecting it. We need to have a consideration of the morphology because it's detecting something that is appreciably and significantly different from the molecular sequence data. This might underscores the point that we need better models for our morphological evolution, because if they are having uncombinable data, then we need better models that are more applicable that might be able to work in these sorts of combined analyses. Quick side note, another part of this is where we might need models for different subpartitions of morphology, where in these couple of papers where I, from 2017, highlighted that there are significant differences in the hard and soft tissues or the dental and non-dental osteological characters in mammals. So like, for example, within mammals, it's the dental characters are less consistent with the morphological, with the molecular trees than the osteological characters are. The osteological characters are showing a better consistency with the molecular trees. So data are important from morphology in this case, but we wouldn't necessarily say that the molecular benchmark has helped us answer that question about which one's better or worse. They both seem to be equivalent, Bayesian and parsimony data. So let's have a look at a different kind of empirical benchmark. You can also evaluate topologies using stratigraphic congruence, as is this example here. These are two topologies of the same group of fossil jawless fish evaluated against the stratigraphic age range data. We can quantify their fit or against this, and we can use that technique to ask this question: which phylogenetic method produces trees that are more congruent with stratigraphic age range data? This is a project with Peter and Emma, who are working in my lab, again with Joe, who I've mentioned. We took a meta-analysis approach, compiling a large number of morphological data matrices from using, amongst others, Graham Lloyd's public repository. We put filters on those and used age range stratigraphic data from the paleobiology database and an automated approach, and then tried to eliminate those that were too heavily in the duplications. We had a final data set of 167 morphological matrices, which was in the region of 4,000 to 5,000 taxa, and a combined high number of characters. We subjected that to Bayesian analysis and parsimony in Mr. Bayes, and parsimony analysis implied in equal weights, and selected some trees to evaluate against the stratigraphic age range data. First finding, that was using the strat package from R from Bell and Lloyd, where we use that to automate and calculate different stratigraphic metrics of performance. Firstly, most of the data sets, the vast majority of them exhibit stratigraphic congruence, right? These phylogenies are having stratigraphic congruence. That's an important headline to work with. But when it comes to the difference between parsimony and Bayesian, we do find a significant difference with trees from the posterior distribution and the most parsimonious trees in terms of which are exhibiting the congruence. It's the parsimony trees that seem to be more congruent with the stratigraphic age range data than the posterior distribution trees. So that's an interesting and important finding. It leads to a number of questions about why. Why is that happening? Well, first of all, we need to remember that, again, we're living with the fundamental differences in the way the trees are sampled. Those posterior distribution trees are sampling a much broader space than the parsimony trees, but I guess in this instance, that's not working in the same way for the simulations, because the parsimony trees are generally looking like they're more precise and more accurate in this instance. Here's an approach where you might want to move away from that with Wright and Lloyd, where they've tried to use the tree space visualization, looking at the small dots as Bayesian posterior distribution trees and the big dots as parsimony trees in terms of the accuracy and precision in terms of stratigraphic congruence. There's a second reason why they might be different. One hypothesis that we put forward in our original paper was that there might be some data peaking going on. Maybe because morphology is the method that is generally evaluated with parsimony, people might be evaluating their trees before they finished it, going back and trying to make a result that's more consistent with the parsimony result. I mean, I wouldn't want to suggest that anyone would look at their phylogenetic data before they finish compiling all their characters. I'm sure none of us have ever done that, but in reality, I'm sure that might be happening sometimes, in which case it might elevate the parsimony, because that's the method that was used to evaluate that tree. So we tried to look under the hood by having a look at the tree samples individually to see what's happening. This is the trees from a Bayesian posterior distribution from one particular data set. And here we tried to consider how the relationship between the posterior probability and the stratigraphic congruence. In this particular example, there's no correlation. The Spearman's row is close to zero. There's no relationship between the probability of a tree or the gap excess ratio. When we expanded that to more data sets, we see a similar pattern once again. There's no relation. We might have expected the more probable trees, the trees with the higher probability, to be more stratigraphically congruent, but we just see no relationship there whatsoever. When we look at the parsimony, however, so this is no longer just the most parsimonious trees. This is a broader tree sample of fully resolved trees that makes it more comparable to the Bayesian posterior distribution. We see, however, there are generally some significant correlations between parsimony score and the stratigraphic congruence. So this is sidestepping this issue of the fact that they're sampling different tree samples, because now we're taking a much bigger tree sample for both, and the level of circularity, because we're not looking at just the parsimonious trees. So when we do this, we find that actually there is no particular strong relationship between the posterior probability of a tree and its strength with stratigraphic congruence, but there is for the parsimony score. The shorter trees are the ones that demonstrate a stronger correlation with the stratigraphic age range data and are more consistent with the independent stratigraphic age range data. So that's an interesting finding. This may have implications for methods which simultaneously try to derive topologies using both morphology and stratigraphic age range data. So I realize I'm running out of time, so I've gone a bit quickly, but nevertheless, I should try and wrap it up. Looking to the conclusions for all three aspects of this talk, both the simulations and empirical benchmarks, the stratigraphy and the molecular frameworks both find significant differences between the Bayesian and parsimony inferences. But this is mostly related to tree sampling. When we eliminate the way that we and compare like for like, they seem to be much more similar to each other. So maybe we don't need to panic too much about the type of method that we're using. We do, however, I would recommend better models of morphological evolution that incorporate selection or the interdependence of characters. We need to understand what the real world properties of morphological data are to better enable Bayesian analyses to have models of morphological evolution that reflect reality, much like we might have for molecular evolution. And maybe we should move beyond broad tree metrics and start thinking about the use of visualization rather than looking at just the topology in and of itself. What is the sort of tree space that's being occupied by our results? How does that cloud of results look in this sort of visualization of two-dimensional space? And I thank my funders from the BBSRC and the Paleontological Association, and I best stop at that point.